Assalamu alaikum students, I hope you're all doing fine. So our today's topic is viruses. Before we begin with viruses, we'll just quickly go through what we studied the last time, and we were comparing the prokaryotic and the eukaryotic cell. I hope you're all clear by now on what a eukaryotic cell is and what a prokaryotic cell is, right? And we'll be trying and finishing up this chapter today. So we'll be studying viruses today. As far as the prokaryotic and the eukaryotic cells were concerned, we discussed that prokaryotic cells were probably the first cells that existed on the surface of the earth, right? What are viruses? We did not talk about viruses anywhere. We talked about 5 kingdoms, right? We said that there is one kingdom that belongs to Monera, then there is the kingdom Prokaryote, then there were 5 kingdoms we talked about, right? The first one was Prokaryote, that consisted of prokaryotes. Then we had Protoctista, that had simple organisms, right? So then we had fungi, then we had plantae, and we had animalia. So we have 5 kingdoms, right? Prokaryote, Protoctista, fungi, plantae, and animalia, right? Out of these, we discussed that the first one, the cell is a prokaryotic cell. In all other 4 kingdoms, the cell is a eukaryotic cell, the cell without a nucleus and the cell with a nucleus. That means this kingdom contains bacteria and cyanobacteria, and the rest of the kingdoms or the rest of the cells, they're all eukaryotic cells. The cells of plants, animals, fungi, and Protoctista, they're all what? Eukaryotic cells. We did not talk about on the verge of living and non-living things. Like we say that viruses, in some, like there's a school of thought that considers that viruses are non-living things. Then there's a school of thought that considers that viruses are living things. Why are we not including virus in a prokaryotic or a eukaryotic cell? The basic reason is because viruses do not have a cell. We use a term for viruses that viruses are what? They are acellular. Acellular means they do not have a cell. Now, because they do not have a cell, hence we cannot classify them as prokaryotes or eukaryotes, right? We use the word acellular for them. So viruses are considered here, somewhere here, on the borderline of living things and non-living things, right? Now, when they do not have a cell, that means they do not have a cell membrane. They do not have a proper cell membrane or a boundary or a cytoplasm in them, right? What are their properties then? What do they look like, right? They have different shapes and they have different properties depending upon the type of viruses, right? But what we do know about them is, number one, all of them are acellular, and then we use a word for them that all of the viruses are obligate parasites. What do we mean by obligate parasites? Viruses cannot survive, cannot live, cannot reproduce outside a cell. They need a host cell to feed on, to live on, right? Because they do not have a cell of their own, right? So they live in the cells of hosts. That's why, and the organisms in which bodies they live, they cause diseases in them. Hence, we call them parasites. Now, because they can only exist as parasites, hence we call them what? Obligate parasites. Viruses cause diseases in plants, viruses cause diseases in animals, viruses cause diseases in fungi, viruses cause diseases even in bacteria. Viruses can even attack a prokaryotic cell. So viruses can use prokaryotic cells as their host cells as well. Therefore, we use the term obligate parasites for them, right? The next thing, if they do not have a cell, what do they look like then? what is the structure of a virus, right? So if we talk about the structure of a virus comprises only of two basic parts. There is nothing else present in them. There are only two basic parts of the cell structure or the structure of virus, and the two basic parts are, number one, the genetic material. That may be DNA or they may contain RNA. So they may be DNA viruses or they may be what? RNA viruses, right? If they have DNA in them, then obviously they are DNA viruses. If they have RNA in them, then they are termed as RNA virus. We also call them retroviruses, right? And the second part that they have is a protein coat. That's it. There is no cell membrane, there is no cytoplasm. The only reason we consider them as living things is their ability to divide, their ability to self-replicate. As you all already know, that DNA or RNA is a self-replicating chemical, right? This DNA or RNA molecule is able to replicate itself. Now, because it can replicate itself, hence the virus can replicate itself, right? So, because replication can take place, hence that is the only reason why we call them living things. Otherwise, they have all the properties of non-living things. That's why we do not place them in any kingdom. We have placed them outside the kingdoms. This one reason, and the second reason being because they are acellular. They do not have a proper cell, right? So what is the shape? How is it arranged? How is the protein coat arranged? How is the genetic material arranged, right? So there are different shapes of the protein coat. I'll just draw one for you. Proteins, they fold on their own. And the coat that the proteins, after folding, these are individual molecules, right, of proteins. They join together and they form a coat. The protein coat has a proper name, and the name of the protein coat is capsid. The protein coat of a virus is called a capsid, right? And these individual folded proteins, the units that make up the capsid, these individual units, they're called capsomeres. So these are basically what? Proteins. Folded proteins, individually folded proteins. They come together, different capsomeres come together, and they give rise to what? A proper protein coat known as what? A capsid, right? And then inside the capsid is present genetic material, DNA or RNA. DNA or RNA, the genetic material is present inside the capsid, and this is what a virus looks like. A virus may have different shapes. The protein coat may vary in shape. They may look like this, a tadpole shaped protein coat. Some viruses look like this with a DNA molecule, or they may look like this with a protein coat, and then there is a DNA or RNA inside, right? So this is what basically they may have different shapes, but the basic structure comprises only of two basic things, and that is what? Genetic material and a protein coat. DNA or RNA and what? A protein coat. Viruses were discovered way later than the bacteria was discovered. The reason being their very extremely small size, right? We had filters, and through those filters, bacteria was not able to pass, but viruses could pass through those filters. That is when we realized that there must be some organism that might be able to divide, that might look like something like a bacteria, but the only property that this had in common with bacteria was that some bacteria are also parasitic, and the viruses are also what? Parasitic. Apart from that, it does not share any other property with bacteria, right? Because bacteria have a proper cell, they are prokaryotic, whereas viruses do not have a proper cell. They are what? Acellular organisms, right? So that's all about viruses. I hope it's clear. One more topic that I wanted to touch today was microscopy. We skipped a few pages of microscopy when we started with the cell structure. So what I wanted to discuss today was microscopy as well. So we'll be studying the basics of microscopy. Whatever you need to know for your paper, right? Basically, in the 17th century, so somewhere around 17th century, a simple microscope was invented, right? Then it kept on getting better. But we had a proper microscope only in the 19th century when proper lenses were invented, right? So why was microscopy so important? See, to look at the organisms that were like the macro properties of the organisms, we could look at them with naked eye. But as we all know, that the basis of biology is a cell. Every living organism is made up of a cell, whether a prokaryotic cell or a eukaryotic cell. So we needed to know more about a cell. To study the cell, we needed a microscope. A proper microscope was required, right? So the microscope that was first made was the light microscope. And the light microscope obviously used light, a beam of light, right, to magnify the images, right, to magnify the size of the cell that we were using, and the lenses used to magnify the size of the image that we were trying to view, right? But it had its limitations. The light microscope could not resolve the image. It could not show you the clarity, right? So two terms that I just used, the term that I used was resolution and magnification, right? So there are two properties of microscopes that you need to study before we can study the differences between light and the electron microscope, right? What do we mean by resolution and what do we mean by magnification? So we'll touch magnification first. When you click a picture using your mobile or any camera, right? After the picture has been clicked, when you zoom in or you zoom out, you can only change the magnification. You cannot change the resolution. The resolution is fixed. By zooming in or by zooming out, you're changing the magnification. So what we basically mean by magnification is the number of times the image can be enlarged. That is termed as its magnification, right? The number of times you can enlarge an image using any device, using your mobile or using a microscope, that is termed as what? Magnification. What do we mean by resolution? Resolution is a word that we derive from resolve. It basically, in simple layman term, it means how clear a picture is. The clarity of the picture, that is termed as its resolution. Now, if you move towards more basic details, the greater the resolution, obviously the clearer the picture, right? The ability of a device to distinguish between 2 points, that is termed as resolution, right? The ability of any device, or specifically of a microscope, to distinguish between 2 points, to show them as two individual points, that is termed as resolution, right? Now, this resolution basically depends upon the source that you're using. In case of the light microscope, the source that we are using is like a beam of light, a wave of light, right? And you all know already that the visible beam of light is from 400 nanometer to 700 nanometer. That means even if you had the best lenses available, you could not see images or you could not see details or the size or the organelles that were smaller than this size, right? Now, I would like to add here one more thing. We say that the resolution of the light microscope is 200 nanometer, whereas the visible beam of light is 400 nanometer. Why do we say that the resolution of a light microscope is 200 nanometer? It is using light as its source, right? And the smallest like wavelength that can be seen by light, or the smallest wavelength of light, is 400 nanometer. But we can still see objects as small as 200 nanometer. The reason being, as you already know, that light travels in waves, right? There are waves of light. And I hope you already know the term wavelength, right? An object that can interfere with the wavelength can be seen with the light microscope. So objects which are equal to half of the wavelength, they're able to interfere with light. When they interfere with the wavelength of light, they can be observed using a light microscope, right? Therefore, the visibility or the resolution that is of a light microscope, that is 200 nanometer, half of the wavelength of the visible light, right? So till many years, till the late 19th century, we were okay with the light microscope because it could show us the details of the cell. But we could not see what is present inside the organelles. We had very small organelles, like you say, see that mitochondria is visible as a dark spot in the presence of a light microscope. But 25 nanometer, the size of ribosomes is 25 nanometer. They do not interfere with the wavelength of light. Supposedly, this is mitochondria. Mitochondria interferes with the wavelength of light because its width is about 1000 nanometer, right? Therefore, it interferes with the wavelength of light. But the size of a ribosome is 25 nanometer only. It is not going to interfere with the wavelength. So therefore, it's not even visible with a light microscope, let alone the details, right? So we started working on getting something better to get a clearer, a better picture, right? We talked about X-rays, they talked about infrared rays, even microscopes were made that used X-rays, that used infrared rays, but they were not very successful. Then came in the electron microscope. Electron microscope uses a beam of electrons, a beam of free electrons, that when they get energy, when they get a lot of energy, the electrons are released from a metal rod, maybe, the electrons can be released. There were two properties of electrons that made them ideal to be used in a microscope. First, they had a very short wavelength. The shorter the wavelength, the more is the resolution, the more is the resolving power. Number two, because they were negatively charged. Now, how is this negative charge helping us? Because they were negatively charged, we could use deflecting plates to bombard them onto the cells. We had to prepare cells, obviously, in a special way for the electron microscope, but because the beam of electrons was negatively charged, we could use a positively charged plate to move them wherever we wanted to bombard them, right? So electron microscope, the resolution is about 0.5 nanometer. That means it can show us structures that were not even close to being visible using a light microscope, right? This is about 400 times more than the light microscope, right? So because its resolution was greater, electron microscope showed us a lot of details. There are two types of microscopes that you need to know. Two basic types are light and electron microscope, but then electron microscope has two further types. We have a scanning electron microscope and a transmission electron microscope, right? It is termed as SEM and TEM. Scanning electron microscope and transmission electron microscope, right? I'll just write the word here. T stands for transmission, whereas S stands for scanning. What is the basic difference? The name indicates. Transmission electron microscope uses a beam of electron that has passed through the cell or through the whatever object you are viewing under the microscope, right? So transmitted rays of electrons that have passed through the cell, they are observed in the transmission electron microscope. The difference is more details, right? Here, the reflected beam of, in the case of scanning, the reflected beam of electrons is observed. That means if you have to look at just the surface of something, we will use what? Scanning electron microscope, right? The electrons will touch the surface, they will be reflected, and we'll be studying the reflected rays only, right? In this case, in the transmission, we'll be studying the electrons that have passed through the cell to get more details, to get to know more details of the cell, right? So there are two basic types: scanning electron microscope and transmission electron microscope, right? So I want you to read all of this. It's mentioned in your book. One more thing that you need to know about magnification is how to calculate magnification. You might have done some work on it in your O3 in the ATP paper, but a few details there should be that you need to know. Magnification. Now, there is a question in your practical paper as well, sometimes in your theory paper as well, where you're asked to calculate magnification, right? You're given a diagram and you have to calculate the magnification, right? How do you calculate the magnification? We have a simple formula. Magnification is equal to image over actual, I over A. If any two of these are given, you can calculate the third one. If you are given magnification, you can calculate image. If you're given magnification and the actual value, you can calculate the value of image. If you are given the image, you'll have to measure that image, obviously, and you can calculate the actual value if you're given magnification and the image. So if any two of the values are given, the third value can be calculated. Just a few basic steps that you need to know that how to calculate it, right? So for example, you're given a diagram. There are cells in that diagram. Supposedly this is called, let's say, हम इसको नाम दे देते हैं. This is called figure 1.1, right? And they tell you that the size of this particular cell, L, is equal, the actual size is 8 micrometer, right? Now, you have to calculate the magnification. They ask you to calculate the magnification you need to calculate. You're given the actual size, the size of cell L, actual size of L is 8 micrometer, right? They haven't given you the image, but you have the picture with you. What you need to do is use a scale to measure it. So basically, what is I? I is the size that can be used, size that can be measured using a scale. That is the I. And A is the actual size of the cell. Now what you will do is, you'll take your scale and you will measure this size L, right? Whatever answer you get, let's say this is 5 centimeter. You say the cell L is 5 centimeter. This is the image, right? And the actual size that you have, that is 8 micrometer. The first thing that you have to do is, units आपको same करनी है. सबसे पहला काम जो आपने करना है, right? 1 centimeter is equal to 10 millimeter. Please remember this, write this down somewhere. Whenever you are measuring using your scale, never write your answer in centimeter. You have to measure in millimeter. The next step is to convert this millimeter into micrometer. So 1 millimeter is equal to 1000 micrometer, right? So what I'll do is 5 multiplied by 10, I'm converting it to millimeter. That is equal to how many millimeter? 50 millimeter. Then I'll multiply this 50 with 1000, and that is equal to 50,000 micrometer, right? here's your answer, right? So you've got now both of your values in the same units, right? The actual size is 8 micrometer and you've got your size, that's 50,000 micrometer. All you have to do is now divide 50,000 by 8 and you will get the magnification. This is when you're given what? The diagram, right? I'll repeat this quickly up. To calculate the magnification, the simple formula that we use is magnification is equal to image over actual, right? Image means the diagram that you're given, right? That you can use measuring a scale, and actual means the real size of the cell, right? If any two of these given, the third can definitely be calculated, right? So you were given a diagram called figure 1.1. The actual size of the size L, cell L, was 8 micrometer and you were asked to calculate the magnification. So L is equal to, we measured its image size, that was 5 centimeter. We converted that 5 centimeter into micrometer because the units have to be same to be solved, right? So it came out to be 50,000 micrometer, right? Then we knew the actual size already. Divided image with the actual size, whatever answer you get, that is your magnification. One more thing that might be new for you, sometimes you're given a cell. I'll write it down somewhere here. So sometimes you're given a cell and you're asked and there is a bar drawn under it. And they ask you to calculate the magnification using the scale bar. Let's say they tell you that the actual size is, it would say here, 6 micrometer. Now, this does not mean that this length of this bar is 6 micrometer. This is called calculating magnification using a scale bar. This bar is termed as a scale bar. So we are using what? A scale bar. This was using the diagram and this is using what? A scale bar. This is termed as a scale bar, right? Whatever number is mentioned over the scale bar, that is actually the actual size, right? Now in this case, you do not have to measure this cell. You actually have to measure the scale bar. So when a scale bar is given in the diagram, you will not measure your diagram. You'll actually use your scale to measure this scale bar, right? I'll solve it for you. Let's say you have to find magnification of this. Actual size you're given, that is what? 6 micrometer. Image you have to calculate now. How? Not measuring the cell, you will be measuring what? The scale bar. Let's say the scale bar size was 3.6 centimeter. That means I'll have to multiply it with 10. You will never give your answer in centimeter. You will multiply it with 10 and this will turn out to be 36 millimeter, right? Now, again, you have to have same units. This 36 has to be multiplied with 1000 to be converted to micrometer. That means 36,000 micrometer, right? So how do I calculate magnification? Image over actual, 36,000 divided by 6. Whatever answer you will get here, that is your magnification, right? So this is how you calculate magnification using a scale bar. This is going to help you in practicals as well, and this is going to help you in theory paper as well. So sometimes you're asked to calculate magnification using the actual diagram, and sometimes you're asked to calculate magnification using what? A scale bar. So when a scale bar is given, in that case, you will not measure the width or the length of the image. You will measure what? The scale bar. And whatever is written on the scale bar, that is the actual size, right? I hope I'm clear. One more last thing. Whenever you are calculating or measuring the size of the cell in your practical paper or in your theory paper, especially in your practical paper, what you have to do is, do not measure it once. You have to take average, right? You have to measure it at least thrice, right? And then let's say I measured it for the first time and my answer came out to be 50 micrometer. I measured it again, it was 51, and again it was 50. I'll add all three of them up and then divide them by 3. Whatever answer I get here, that will be my average answer, right? So you never measure once, you measure thrice, and then you take average out of it, right? So this was all for today, and we're done with the chapter. What I want you to do is read light microscopy and read electron microscopy at home. And then if you have any questions, you can ask me tomorrow. Thank you very much.